public health officials and epidemiologists are issuing a sharpened set of warnings regarding the seasonal risks of hantavirus, particularly for individuals engaging in the cleaning of long-dormant spaces such as attics, basements, and sheds. While the virus remains statistically rare, the high mortality rate associated with hantavirus pulmonary syndrome, or HPS, has prompted a series of advisories focused on the mechanism of aerosolization. In the Americas, the primary threat is the sin nombre virus, which is carried by the deer mouse, though other rodent species like the white-footed mouse, the rice rat, and the cotton rat also serve as reservoirs for various hantavirus strains. The current reporting emphasizes that the danger is not merely through direct contact or bites, but through the inhalation of microscopic particles. When rodent urine, droppings, or nesting materials are disturbed, the viral load contained within these materials can become airborne in the form of dust. A single breath of this contaminated air in a confined, poorly ventilated space can be sufficient to introduce the pathogen into the lungs, where it begins its incubation period. This specific mode of transmission makes standard domestic activities, such as sweeping or vacuuming an infested attic, high-risk behaviours. Epidemiological data indicates that many cases occur in rural or semi-rural settings where human dwellings overlap with the natural habitats of these rodent reservoirs. However, the risk is not exclusive to these areas, as rodents frequently seek shelter in suburban structures during colder months. The framing of the current public health response is built around the concept of prevention through awareness, as there is currently no vaccine or specific cure for the infection. The timeline of the illness typically begins with a febrile phase that can easily be mistaken for more common respiratory viruses or the flu. Initial symptoms include fever, severe muscle aches, and fatigue, often localized in the large muscle groups of the thighs, hips, and back. As the disease progresses, usually within 4 to 10 days after the initial phase, the respiratory system becomes the primary site of distress. The transition from mild illness to severe respiratory failure can be rapid, characterized by the accumulation of fluid in the lungs, a condition known as non-cardiogenic pulmonary edema. This progression underscores why officials are focusing so heavily on the initial exposure event in attics and storage spaces. By the time a patient presents with advanced symptoms, the window for effective supportive care narrows significantly. This report will examine the specific mechanics of this viral threat, the current state of laboratory surveillance, and the rigorous protocols required to mitigate risk during cleaning and maintenance activities in high-risk environments. The clinical and laboratory response to hantavirus requires a highly coordinated effort between local healthcare providers and national reference laboratories. Because the symptoms of hantavirus pulmonary syndrome overlap so heavily with other viral syndromes during the prodromal phase, diagnostic confirmation relies on specialized testing that is not available in most standard clinical settings. State health departments and the Centers for Disease Control and Prevention typically manage the definitive testing, which involves enzyme-linked immunosorbent assays to detect specific IgM and IgG antibodies. In some cases, polymerase chain reaction, or PCR, testing is used to identify the viral RNA in clinical samples. The core development in current surveillance is the monitoring of rodent population dynamics, which are often influenced by environmental factors such as mild winters or high rainfall that leads to an abundance of food sources. When rodent populations surge, the likelihood of human-rodent interaction increases, particularly in enclosed structures. Medical agencies emphasize that there is no person-to-person transmission for the strains of hantavirus found in North America, a critical distinction that separates it from other high-consequence pathogens like the flu or COVID-19. However, the Andes virus in South America has shown a limited ability for human-to-human spread, which keeps global health monitors on high alert regarding the evolution of these viral families. In hospitals, the focus remains on aggressive supportive therapy. Because there is no FDA-approved antiviral treatment specifically for hantavirus, patients in the late respiratory stage often require admission to an intensive care unit. This frequently involves mechanical ventilation and, in the most severe cases, extracorporeal membrane oxygenation, or ECMO, to oxygenate the blood while the lungs are compromised. Clinical reports indicate that once the leakage phase begins, where capillaries in the lungs allow fluid to seep into the air sacs, the survival of the patient depends almost entirely on the quality of the supportive interventions and the timing of their delivery. Research labs are currently investigating potential therapeutic pathways, including the use of neutralizing antibodies and various broad-spectrum antivirals, but these remain in the experimental stages. The infrastructure for monitoring hantavirus is also linked to broader biosafety concerns. Handling the live virus requires biosafety level 3 or 4 containment, which limits the number of facilities capable of performing primary research. This logistical constraint means that much of our understanding of the virus comes from retrospective studies of confirmed cases and field surveillance of rodent hosts. Officials state that the primary challenge in managing an outbreak is the silent nature of the transmission. A homeowner may inhale the lethal dust weeks before they feel the first symptoms, making it difficult for clinicians to establish a clear history of exposure unless specifically prompted to ask about recent cleaning of outbuildings or attic spaces. Prevention and public guidance remain the most effective tools in reducing the incidence of hantavirus infections, as the environmental stability of the virus is relatively short-lived when exposed to sunlight and disinfectants. Public health agencies are adamant that the traditional method of cleaning dusty areas, sweeping or vacuuming, must be avoided if there is any evidence of rodent activity. Instead, a wet cleaning protocol is the international standard for safety. This involves saturating the area with a disinfectant, such as a 10% bleach solution, for at least 5 minutes before any cleanup begins. By wetting the droppings and nesting materials, the risk of aerosolization is neutralized, preventing the virus from becoming airborne and inhalable. For individuals working in high-risk environments, such as professional cleaners or homeowners dealing with significant infestations, the use of personal protective equipment is strongly recommended. This includes non-porous gloves, long sleeves, and, most importantly, a properly fitted N95 or higher-rated respirator. It is a common misconception that a standard surgical mask provides adequate protection against the fine particles that carry hantavirus. Only respirators capable of filtering out submicron particles are considered effective. Surveillance of residential and commercial properties should also include proactive rodent proofing. This involves sealing any gaps larger than 1/4 of an inch, which is small enough for a mouse to squeeze through. Officials also suggest removing potential food sources and nesting sites from the immediate perimeter of the home to discourage rodents from entering. Despite these clear guidelines, significant gaps in knowledge remain. Epidemiologists are still studying why certain individuals exposed to the virus develop fatal respiratory failure, while others may experience a more mild form of the disease. There is also ongoing research into the long-term health effects for survivors of HPS, as the intense pulmonary stress can lead to lasting changes in lung function. Global health organizations continue to monitor the impact of land use changes and climate patterns on the geographic distribution of hantavirus, as shifts in rodent habitats could bring the virus to new, unprepared populations. The overarching message from health authorities is one of cautious prevention. While the risk of encountering the virus is low, the consequences of a single breath of contaminated dust are high enough to warrant strict adherence to safety protocols. By treating every neglected attic or shed as a potential biohazard, individuals can effectively manage their risk and prevent the onset of this severe respiratory illness. For more updates on public health surveillance and global reporting, consider subscribing to this channel. The clinical trajectory of hantavirus infection is defined by a deceptive onset followed by a rapid, often catastrophic, respiratory decline, which places an immense burden on diagnostic labs and intensive care units. Once the viral particles are inhaled from disturbed attic dust, the pathogen targets the endothelial cells that line the body's blood vessels, particularly within the lungs. Medical reporting indicates that the incubation period can range from 1 to 8 weeks, though most patients begin showing symptoms within the first 3 weeks post-exposure. This delay often obscures the link between a specific cleaning event and the subsequent illness, complicating the initial triage process in emergency departments. During the early or prodromal phase, the symptoms are indistinguishable from common influenza or other seasonal respiratory viruses, characterized by high fever, severe myalgia, and gastrointestinal distress. However, public health officials warn that the lack of upper respiratory symptoms, such as a runny nose or sore throat, can be a clinical red flag for hantavirus when coupled with a history of rural or domestic rodent exposure. As the virus replicates, it induces a systemic inflammatory response that leads to capillary leak syndrome. This is the physiological hallmark of hantavirus pulmonary syndrome, where the microscopic vessels in the lungs begin to leak plasma into the alveolar spaces. This transition from the prodromal phase to the respiratory distress phase can occur in a matter of hours, often referred to by clinicians as the crash. At this stage, the patient's oxygen saturation levels drop precipitously, and the heart must work under extreme pressure to compensate for the fluid-filled lungs. Laboratory confirmation remains one of the most significant hurdles in managing the spread and treatment of the virus. Because most community hospitals lack the specialized equipment to test for hantavirus, blood samples must typically be couriered to state-run public health laboratories or the Centers for Disease Control and Prevention. The primary diagnostic tools are serological assays designed to detect IgM and IgG antibodies specific to the virus. In the early stages of the disease, an IgM-positive result is a definitive indicator of an acute infection. Additionally, molecular testing using reverse transcription, polymerase chain reaction, or RT-PCR, can detect the viral genome in the blood or tissue samples, though this is often more effective during the peak of the febrile phase. For cases that result in fatalities before a diagnosis is reached, immunohistochemistry is performed on lung tissue during autopsy to visualize the viral antigens within the capillaries. The logistical delay inherent in shipping these samples to reference labs means that clinicians must often make high-stakes treatment decisions based on clinical suspicion alone. This is where the one-breath warning from epidemiologists becomes a critical piece of the patient's history. If a doctor is made aware that a patient recently disturbed a significant amount of dust in an enclosed shed or attic, they are far more likely to initiate the aggressive supportive care required to stabilize the patient before the results of a lab test return. In the absence of a targeted antiviral medication, the standard of care is strictly supportive, requiring the highest level of medical infrastructure. Patients who progress to the respiratory stage of hantavirus pulmonary syndrome almost universally require admission to an intensive care unit. Statistics from previous outbreaks show that early intubation and mechanical ventilation are essential, but even these measures may be insufficient if the pulmonary edema is severe. In these critical instances, hospitals may employ extracorporeal membrane oxygenation, or ECMO. This advanced technology temporarily takes over the function of the heart and lungs, pumping the patient's blood through an artificial lung to remove carbon dioxide and add oxygen before returning it to the body. This provides the patient's own lungs the time necessary to clear the fluid and recover from the viral assault. However, ECMO is a resource-intensive therapy available only at major medical centers, creating a significant disparity in outcomes for patients in remote or rural areas where the virus is most frequently encountered. Furthermore, clinicians must manage a concurrent phase of cardiovascular instability, characterized by low cardiac output and hypotension. Unlike many other forms of respiratory failure, the shock associated with hantavirus is particularly difficult to treat because the fluid leakage makes traditional intravenous fluid resuscitation dangerous, as it can worsen the pulmonary edema. Surveillance efforts at the state and federal levels serve as the final line of defense in identifying emerging clusters of the virus. Epidemiologists track confirmed cases to identify geographic hotspots where rodent populations may be carrying a higher than average viral load. This data is often cross-referenced with environmental studies that monitor the prevalence of the virus in local deer mouse populations. When a case is confirmed, public health agencies often conduct field investigations to trap and test rodents in the vicinity of the patient's home or workplace. This active surveillance helps officials determine if a particular setting, such as a specific shipping facility, a construction site, or a residential neighborhood, poses an ongoing risk to the public. While the source material focuses on the extreme risk of domestic dust, global health organizations also monitor the potential for the virus to impact trade and logistics, particularly in the agricultural sector, where large grain storage facilities can become epicenters for rodent activity. Despite the high mortality rate, health agencies emphasize that the virus is not a resilient pathogen outside of its host. It is highly susceptible to common detergents and heat. The challenge lies not in the hardiness of the virus itself, but in the environment where it is found. The poorly ventilated, dark and dusty corners of human habitation where a single lapse in precaution can lead to a life-threatening exposure event. Given the absence of a vaccine or targeted pharmaceutical intervention, public health agencies emphasize that the only definitive protection against hantavirus is the rigorous application of prevention and exclusion protocols. The primary practical takeaway for any homeowner or maintenance professional is the absolute prohibition of dry cleaning methods in areas with suspected rodent activity. To prevent the aerosolization of the virus, a wet-down decontamination procedure is the standard requirement. This involves saturating any area containing rodent droppings, urine, or nesting materials with a disinfectant solution, typically a mixture of 1 part household bleach to 9 parts water. Officials stress that the solution must remain in contact with the contaminated material for at least 5 minutes to ensure the virus is fully neutralized before it is disturbed. Only after this soaking period should the material be wiped up using paper towels or a mop. Furthermore, the disposal process is equally critical. All waste should be double-bagged and removed from the premises immediately. The use of vacuum cleaners or brooms is strictly discouraged, as these tools are designed to move air and fine particles, which is the exact mechanism that triggers the inhalation of the lethal dust epidemiologists warn about. For those entering confined spaces like attics or crawl spaces, the ethical and professional consensus is to treat every environment as potentially infectious. Personal protective equipment, specifically N95 or P100 respirators, is non-negotiable. Public health guidance clarifies that standard cloth or surgical masks are insufficient for filtering the microscopic viral particles. High-risk activities also require the use of rubber or nitrile gloves and long-sleeved clothing to prevent any secondary contact or accidental transfer of the virus to the face. Beyond immediate cleanup, the long-term strategy for risk mitigations involves aggressive structural exclusion, often referred to as building out the rodents, because a deer mouse can enter a structure through a gap as small as 1/4 of an inch, rarely the width of a pencil. The surveillance of a building's exterior must be meticulous. Practical guidance includes the use of steel wool or wire mesh, which rodents cannot chew through, to plug gaps around pipes, vents, and foundation cracks, followed by sealing these areas with cork or expanding foam. Outside the home, the removal of potential habitats is a core pillar of public health advice. This includes clearing tall grass, weeds, and brush piles within a 30-foot radius of the structure and elevating wood piles at least 12 inches off the ground. By reducing the carrying capacity of the immediate environment, homeowners can significantly decrease the probability of a rodent entering the indoor spaces where aerosolization risks are highest. In commercial or shipping settings, these same principles apply but on a larger scale, requiring integrated pest management systems that focus on monitoring and exclusion rather than relying solely on reactive trapping, despite the clarity of these prevention methods. Significant limits to our knowledge persist, creating a landscape of uncertainty that epidemiologists are working to navigate. We still do not fully understand why the virus remains dormant in rodent populations for years before suddenly surging in human incident clusters. There is also an ongoing scientific debate regarding the influence of biodiversity on hantavirus risk. The dilution effect hypothesis suggests that a more diverse ecosystem of non-host animals might actually lower the risk to humans by competing with the primary rodent reservoirs. Furthermore, the long-term physiological impact on the survivors of hantavirus pulmonary syndrome remains a critical area of study, as the severe scarring and inflammatory damage to the lungs may lead to chronic respiratory complications that are currently underreported. As we monitor the shifting boundaries of these viral reservoirs in a changing global climate, The ethical responsibility of public health reporting is to provide clear, actionable information without inducing unnecessary panic. While the virus is rare, the severity of its impact demands a high degree of civic and personal vigilance.